Welcome back to Coordinating Clinical Trials. I am Steve Pope, your instructor, and this week we're going to be discussing data collection, which is going to be probably the bulk of what you're going to do in your clinical research coordinator role. So let's recap things for a little bit here. So first of all, the purpose of a clinical trial is to collect data on a medical intervention that shows efficacy and safety of the intervention in a patient population. So basically how this applies to your role is that you're going to conduct subject visits and then you're going to collect data during those visits based on the assessments that you do at the visits, and we're going to discuss those in a little more detail as we go along. And then you're going to enter that data into a clinical database that will be used for submission to regulatory authorities, such as the FDA, in support of a regulatory approval by the sponsor company. So this is going to be a very key role in the overall process of getting this medical intervention approved. So let's take a look at our learning objectives for this lesson. We're going to discuss the data entry process. We're going to cite the various types of data that will be collected and entered, define what good data quality is, practice entering data, and identify some common errors in data entry. So let's talk about data collection. So we want to begin by talking about what the case report form is. So the case report form is the instrument used to collect data in the clinical trial. It can be in paper form or it can be on what's known as an electronic data capture system, which there still are some paper trials out there, but they're few and far between these days. So you're probably going to be working with an electronic data capture system where you're entering data directly into the clinical database. So the case report form gets its name from literally having a form back in the old days that the coordinator would use, probably put it on a clipboard, and then when the subject came in, they would record the values for each assessment. Maybe they're doing vital signs or height and weight, things like that, and they just write it down on the form, and then that got sent in and somebody at the pharma company entered it into the database. Well, now the process is a little bit different. So you're still going to collect the data in sort of the same fashion in many cases, but you're going to be the one entering it into the electronic data capture system. So you've also got additional pieces of data as well. So you're going to have data that gets collected on what's known as a source document. That could be a medical record from another institution. It can be a lab report. It can be a procedure report that you might have that's done at your clinic. And then you're going to transcribe that data onto the electronic case report form, and then that gets submitted to the sponsor of the trial. Your monitor or your clinical research associate is going to come out to visit your site on a periodic basis, and what they're going to do is review the CRF data that you've entered against the source data where you collected it from the patient visit. So this process is called source data verification, and it's probably the task that consumes most of the monitoring or most of the time dedicated to the monitoring visit, and it's probably going to be the thing that you're going to be involved in for the most part. So what is data quality and how is it achieved? What this means is data that is collected has to be supported by medical documentation. And it needs to be, these are clinically logical and accurate. So for instance, if you have an electronic medical record and your subjects come in, you may have an order set up that corresponds to your trial, so it prompts you to collect the data necessary at that particular visit for the trial. Or you might be using a paper document to collect data. Either way is fine. Um, you're probably going to do a little bit of each in your career as a clinical data coordinator or clinical research coordinator. Um, and so what's going to happen is once you collect that, then as I mentioned before, the CRAs are going to come out and they're going to verify it. They're going to do their source document verification. Also, once you enter data into the database, the medical monitors who work for the pharma company, they're going to start reviewing key patient data too, and they may issue some queries to have you clarify things related to the entry data. You've also got the data management team, so they'll have a team of people that they look at the data and they're going to go through what they call a cleaning process. So they're going to, send queries out to you to have you verify certain clinical data, such as comparing data points like adverse events, medical history, ConMeds, and we'll talk about these in a little greater detail as we go along. So let's talk about what systems we're going to be using to collect the data. As I mentioned, you may be using a paper case report form where you collect the data on paper, but in most cases you're going to be using an electronic data capture system, which puts it directly into the clinical database. So you may be using something like Medidata Rave or Data Labs or Oracle Clinical. There are a number of them out there that you may be using, so you're going to become familiar with it. So what are the expectations of the sponsor? What do they want us to do? Well, they want you to collect data at each subject visit and then enter that as soon as possible into the EDC system. Usually there will be some metric for what their expectation is. Maybe it's within 5 business days, for instance. But once you collect it, you want to enter it into the EDC system as soon as possible. You also want to be able to understand any good clinical practice issues that are related to data review. Those are contained in 21 CFR Part 11. And for most part, your EDC systems are going to comply with that. You also want to understand what data is most important for the final statistical analysis. So when you get your protocol from the sponsor, a portion of that protocol is going to discuss all the fancy statistics that they're going to use to evaluate how effective their data is. So you probably want to be familiar at least with that to understand what the primary endpoints are going to be, and those are going to be listed in the protocol document. So let's start talking about what type of data you're going to collect during your visit. So initially, you're going to begin by collecting some demographic data. This is going to include the subject's age, their gender, what their race and ethnicity is. And this is going to be used to provide analysis on different groups and their association with the study data and the results. You'll collect the date that they signed informed consent. It's very important that the informed consent date needs to precede the date and time of any trial-related procedures that you're going to do. Every once in a while, there could be something related to inclusion and exclusion that might be waiverable, or you might seek permission from the study's medical monitor to enroll a subject. That might be something that's included in your demographics data. And then normally you'll have some queries fire as you start to enter that and we'll talk about those as we move along. In many cases, the demographic data is probably going to be entered into an IWRS system, which is an interactive web response system, which is different from your clinical database. And then that IWRS system will feed the data into your EDC system. So we'll talk about that as we go along. So for demographic data, some of the common errors that you're probably going to see are going to be things like informed consent date is after the screening visit date. That's going to probably cause a query to fire. Date of birth is not within the age range for the study. That's going to cause a query. You may have more than one race checked. That may or may not cause a query. You might check that the patient is female when in fact you're recruiting for a male-only study and vice versa. That's something that would cause a query to fire. That you can easily fix that. So inconsistencies and conflicts are going to cause system queries to fire, and you're going to have to answer those before you can proceed. So you're going to collect inclusion-exclusion data, which is going to be against whatever the inclusion-exclusion criteria is for your protocol, for your study. And that criteria is going to be listed in the protocol. So what I would highly recommend is that you take that inclusion-exclusion criteria, print it out, put it in the subject's chart, and as they come in, you determine whether or not they meet the inclusion-exclusion criteria. So all inclusion criteria should be answered yes, and all exclusion criteria should be answered no. For certain responses, you may get the approval of the medical monitor to enroll that subject. What you also want to do is your principal investigator needs to confirm the eligibility criteria for each subject. So they need to do that in writing somehow. If you're using paper source documents, then you should have that PI sign the inclusion-exclusion criteria page with their date and signature validating that subject meets all of that criteria. If you're working with an electronic medical record, the PI should write a progress note in the electronic medical record indicating that subject meets the eligibility criteria for the trial. So when your subject first comes in, you're going to take the medical history information for that subject. So you may have, for instance, a form that's used to collect that with. So this is going to be information about their medical history, and you're going to ask them a series of questions about whether or not they've had certain diagnoses in the past. So anything that's occurred up to that point is considered medical history. So that's going to establish what their health baseline is at the beginning of the trial. And you're going to enter that into the EDC database on the medical history page. So it's going to be a log page, which means that it's probably going to look like it has rows in it. And it may be, they're going to be pages that you will expand as the trial goes into other stages. So these log pages will be your medical history page, your adverse events page, your concomitant medications page, IP accountability, and medical procedures. So those are going to expand. They're called living pages, so they change over time. So once the subject comes in, say for their screening visit, that data, once you collect that, it's going to remain static at that point. Then they're going to come in for whatever their subsequent visit is. So some of the errors or inconsistencies that are likely to appear on medical history pages might be, illegible entries if you're using a paper document, misspelled entries, those could occur on either paper or EDC. I've listed a couple of examples there. Hypernatremia versus hyponatremia. They're two completely different items there. And so that sometimes can create some inconsistencies. Incomplete medical history. Sometimes you're going to have findings from a physical exam or lab results that may not be recorded until you get the lab result back, for instance, or you may have a different term that may need clarification. Some of the other problems that we see on medical history is you've got incomplete or missing start and stop dates. It's fine if a diagnosis is ongoing, for instance, for say a chronic disease that would likely be ongoing, so there would not be a stop date for those. And then we generally see a large number of entries recorded under one body system. That can flag and be an inconsistency. So we want to take a look at those, and that can sometimes be a very common error. And then finally, medical history that's listed on a concomitant medication page but is missing from the medical history page. It's difficult to do that with an EDC system, because as you enter that, it's going to prompt you for the corresponding number of that medical history diagnosis. So when we go through our data entry exercise, it'll be a little more apparent to you, and it'll make sense a little bit more. So for most trials, when the subject comes in for their visit, the PI or the sub-I is going to do a physical exam. So this is an examination of the body for purposes of diagnosis and treatment. So this data becomes part of the patient's medical record. This data is the physician's assessment of each body part, and it's broken down by body part on the CRF page. So it's important to understand this can only be done by a medical practitioner, an MD, a DO, a physician's assistant, or a nurse practitioner, depending on what state regulations require. Now your protocol may require that only the principal investigator can do physical exams. If that's the case, then only the principal investigator can do those. Some common errors that we see on the physical exam page is illegible entries. Can't quite make out what it is. Entries that are recorded in different body systems on different visits. For example, a respiratory infection recorded in respiratory and then in ear, nose, and throat at a different visit, or abnormalities that should be recorded in medical history. For example, a surgical scar that's noted on a physical exam but is not noted in medical history. That can be a common error. Abnormalities which might be an adverse event but are not listed on the adverse event page. For example, maybe a new skin rash that's observed on a physical exam, but there's no corresponding adverse event. So you have to be careful about those. You may need to go back and talk to your principal investigator about these and clarify whether or not that needs to be added to, say, an adverse event page, for instance. So another log page that you're going to enter on a consistent basis with each subject is going to be an adverse event page. So this is going to look like a log with rows on it. So an adverse event is going to be any untoward medical occurrence associated with the use of a drug, whether or not it is considered drug related. So any adverse change in the patient's health that occurs while the patient is participating in a trial needs to be collected by you. So any event that's considered serious must be reported to the sponsor within 24 hours of the site's learning of the adverse event. And then those will be flagged when you enter the EDC data that it is a serious adverse event. Then investigators must also report serious adverse events to the IRB. So you're going to be involved in that process because you're going to be the one reporting it. Now, there could be certain IRBs certain serious adverse events do not meet their reporting criteria, and that's fine. Probably good practice is for you to report it anyway and then let them come back and let you know that it's not a reportable item. Make sure you keep an acknowledgement that you received from your IRB, because your CRA, your monitor, is going to want to see that when they come out to monitor your sites. And of course, all adverse events need to be recorded regardless of whether they're serious or not. Adverse event is anything from a headache to a heart attack and everything in between. So any change to the patient's baseline is going to be considered an adverse event. So when they come in for their visits, you're going to need to talk with them and see how they've been since the last time you saw them. You can't just ask them, hey, if you had any adverse events, because they don't know what that means. But you're going to prompt them, hey, have you been sick? Have you had to go to the doctor? Have you had to take any medication since you were last here? Those types of questions to prompt that information out of them. Some of the common errors that we see on adverse event pages, very similar to the medical history page. Start and stop date inconsistencies. They don't make logical sense. Ongoing is indicated, but a stop date exists. If you enter that way into the EDC, it's going to cause a query to fire. Inconsistent naming of ConMed pages. Going back and forth between generic names and trade names. That can lead to errors. Missing AEs, lab values that may indicate an AE, but no AE is recorded. Just because you have an out-of-range lab value doesn't necessarily mean that it's an AE. If it is clinically significant and has been assessed that way by the principal investigator, then it needs to be entered as an adverse event. But out-of-range lab values by themselves don't necessarily mean it's an adverse event. And then procedures that are listed instead of medical indications, such as colonoscopy. Well, the colonoscopy is not the adverse event. The adverse event would be whatever prompted the need to do the colonoscopy. So that's something you need to, question your subject. So concomitant medications, these are going to be all the medications that the subject is taking during the trial. Depending on how they've got the protocol set up, they may want you to collect ConMeds from as far back as 90 days before they started the trial. So you may see this listed as both prior and current concomitant medications. So this is a listing of all the meds that the subject is taking. Regulatory authorities use this data to understand the efficacy and safety effects of the investigational product in relationship to other drugs. So it's very important that you get this data as accurate as possible. Common errors that are associated with the concomitant medication page are things such as illegible names of medications. You can't quite make out what it is. Start and stop dates do not match either the medical history entry or the adverse event entry. It's important to understand that every concomitant medication has to be matched up with either a medical history or adverse event entry, unless it's given for some sort of anaphylaxis basis. Another error is dose and dose units not understood. indications not listed on either the medical history or AE pages. You can also have missing data. So these are some of the common errors that we find when entering concomitant medication data. So you're also going to record the study medication, which is the investigational product that you're studying in your trial. So this is going to be recorded for all study medication taken by the subject, and it's going to include the date, time, and dose unit. So these are going to be entered on the investigational product accountability pages. Now you're also going to collect this in your source documents because this is going to be a critical piece of data that the monitor is going to want to see when they come out for a monitoring visit. Some common errors related to study medication include missing data, date and time discrepancies. You may have a trial where the subject records their dosing information in a diary. It might be either on paper, or they might use one of the little ePro devices, which are similar to a smartphone, to collect that. And since they're entering that data, they sometimes make some mistakes on it. So you can have some of these discrepancies that occur as a result of that. So in your role as the coordinator, you're also going to collect study medication compliance. So as you collect that data, you're going to record the study drug was dispensed to the dispensing information for each subject, and then this usually captures the date, time, and the amount of drug that was dispensed, and then what they returned when they came back. This data is usually going to be used to calculate if the subject was taking sufficient medication to be considered a protocol compliant analyzable subject. So they need to be within a certain compliance level, and that's going to be specified in your protocol. Usually it's going to be greater than 80% compliance. So if they have 30 days between visits and you dispense drug to them at visit 1, and then they come back 30 days later, maybe they have a tablet that they take each day, and they come back and they've got 10 tablets left, then they probably didn't take enough of the drug during the in-between visits, so they probably would not be compliant with that. So you're going to check that, and that data is going to be recorded on your study drug or investigational product accountability log. So some of the common errors that we find in the study medication compliance are things like medication numbers dispensed, taken, and returned don't tally. For instance, you record an amount on the IP accountability log, but then the monitor comes out and they're going to check that, and they're also going to check what was returned. You may have a bottle that has pills in it, for instance. They're going to open up that bottle and count the number of pills that are there and make sure it matches what you put on the accountability log. So that's a fairly common error that occurs from time to time. Dates of when the medication was dispensed and returned are not logical, or they're not within acceptable ranges. They don't match the visit dates, for instance. Also, the amount of medication dispensed for a visit is not as per the protocol. So normally what's going to happen is you're going to be working with an IWRS system, and it's going to tell you what to dispense to the subject. Probably will give you a kit ID, and so you go pull that from the inventory, and then you hand that to the subject when they come in for their visit, and that's how you dispense the IP to them. So you need to be familiar with the drug labels that are likely going to be on your IP boxes or bottles or whatever it is you give to the subject. So these labels are going to be stickers from the dispensed drugs that are attached to the CRF, they provide clear proof of the details of the drug dispense and to which subject. So there may be the drug that's on the bottle, you may peel that off and put it on a piece of paper, for instance, and then it has the drug ID, the bottle ID number on it, the kit ID that is, and then that identifies which one you dispense to the subject. So the study medication compliance page details what normally would have some of the information of these labels. And then drug labels contain the randomization number and are very important that these match those recorded in the CRF, as this information tells us what medication the subject was actually taking. So some common errors that we see related to drug labels. Labels are sometimes missing for the dates when the drug were dispensed as per the drug compliance page. Multiple labels are pasted in the same box provided on the CRF for a single label. They might get mixed up from time to time, where you put the wrong label on the wrong piece of paper. So those are some of the common examples of errors that you might see. So one of the tasks that you might do as a coordinator is that you might create some of the source data documents that you're going to use to collect the trial data, the subject data. These can be paper source documents that are used to collect the data. You might use, Microsoft Word, for instance, to create a form that's used, or you might use some other tool. There are tools out there that will do that. If you're given that task, you have to be sure to follow the schedule of events in your protocol so that you make sure that you're collecting, each piece of data that you're supposed to collect at each visit. It's also important to understand these cannot be copies from your EDC system. So most of the EDC systems now, you can print out a page of data, say for each visit, but you can't use that as your data collection tool. Another thing that's more and more commonplace is that sponsors will usually no longer provide paper source documents to sites. That used to be fairly common years ago, but they've gotten away from that because they don't want to somehow influence or introduce some kind of bias into the data collection process. So in all source data, it doesn't matter if it's on paper or if it's in an electronic medical record, it needs to adhere to the ALCOA principles. So ALCOA is just an acronym. It stands for attributable, which means it identifies who documented the data. It needs to be legible, so the data is readable and the signatures are identifiable. Contemporaneous, data is recorded at the time it was collected, not a week or so later, for instance. Needs to be original, primary source data. This could be a printout, say from a lab report, or it might be from an observation. So if you're, say, taking vital signs, for instance, you've observed those vital signs and you've recorded that on your source document. It also, needs to be accurate, so free of errors, and if any editing should occur, it needs to be, if a change is made, it needs to be struck through and then signed or initialed and dated by the person who struck through it. So back in 2010, these ALCOA principles were expanded to include a few other components, and we refer to this as ALCOA Plus. So these are complete, which means that all data, including metadata and audit trails, is there. C is for consistent. The data is chronologically arranged with some type of time stamp. And then it's enduring, so data is stored securely and long term. And then finally available, which means that data is accessible whenever needed. It's not locked up in some filing cabinet someplace where no one can get at it. So all those four areas include the Alcoa Plus principles. There are several reading assignments that are going to amplify what I've gone over in this lesson. You've got chapter 10 from your textbook, which is case report forms and electronic data capture. Also got a couple of articles there that discuss data collection and that task for coordinators at sites. So I want you to read through those, and that'll help expand on what I've gone over in this lesson. So finally, for your discussion forum assignment for this lesson, imagine that you're a clinical research coordinator working at a site. You're working on a clinical trial for a new treatment of hypertension. So what I've done, I've put two documents out there for you. One is going to be the source documents of, they're going to be in the week 9 folder on Blackboard. So you can pull those up. It's going to be a zip file. So there's several worksheets in there that comprise the source documents for a subject visit. And then I want you to enter the data into an EDC system. So I'm using just a simple Excel spreadsheet. And when you open that up, some of the data has been entered. So there are some empty cells in that spreadsheet. They are shaded gray. So I want you to enter that data from the source data documents that I put out there on Blackboard. And then once you've done that, to complete the exercise, I want you to post the spreadsheet in the discussion forum. Then I'll take a look at it and I will wrap everything up at the end of the week.